चलो राजू से यूनिवर्सिटी 122 Good morning to all participants. We are supposed to start the program in another 2 minutes. So be there. I appreciate your patience waiting. Thank you. सर को फोन कीजिए.
Hello. Hello.
Sir, we'll start now 1:50.
Hello. Hello.
नहीं आ रहा सर आपका. Your voice is not coming. अब ब्लॉक नहीं दो. नहीं कहाँ पे है आपका सर? No, in this one where is it? Ask to now unmute कीजिए सर. Unmute yourself. हाँ.
हो गया?
हाँ.
Hello.
Yeah. Audible.
Sir, good morning. Raman sir.
Yes, sir. Good morning.
Good morning, sir. Now
Shall we start,
sir?
sir?
There was some problem.
Yeah, please start.
Shall we start?
Yeah, please start, sir.
Mandar,
Yes.
are you ready?
Yes, absolutely.
Okay. Sujit?
So good morning to all participants. I, Sujit Kumar, on behalf of Honorable Chancellor of KD Academy of Higher Education and Research, Dr. Prabhakar V. Kore sir, Honorable Vice Chancellor Vivek A. Sahu ji sir, Honorable Registrar Dr. V. A. Kotiwale sir, Head of the Organizing Committee Professor Dr. Raman Dan sir, the Principal of KD College of Pharmacy, Bangalore, Convener of today's webinar Dr. Suhas Karti sir, the project manager of DSRC of campus Bangalore, and I Sujit Kumar, assistant professor in Department of Pharmaceutical Chemistry at Kelly College of Pharmacy Bangalore, and coordinator of today's webinar on an Reaxys, an innovative data analytical tool by Dr. Mandar Bodas, Elsewhere Solution Sales Consultant, and Lavanya Trika, account manager Elsewhere. Welcome all participants for this wonderful webinar. Friends, before we go for the webinar starting, I would like to request our convener of today's webinar, and project manager of BSRC of campus, Dr. Subhasis Karki, to introduce the speaker of the today's talk. Over to you, sir.
Good morning, all delegates and participants. And it's an honor to introduce our today's webinar presenter, that is solution consultant of Elsewhere, Mandar Bodas, doctor. And from September 2018, he is working as a solutions consultant in Elsewhere. And before that, he was there in Wycon Bristol Manuscript, Bangalore, as a senior principal investigator from 2008 to 2018. And he worked on therapeutic areas, especially neuroscience, atherosclerosis, heart failure, and fibrosis areas. And earlier to this, he was there in Ranbaxy Laboratories, therapeutics area, as a research scientist. Area is new drug discovery research, especially with oncology, combinatorial library synthesis from 2006 to 2008. And he has done postdoctoral studies at the University of Montreal. And area is research area was solid phase peptide synthesis from 2004 to 2006. And he did PhD, the Doctor of Philosophy from NCL, Pune, title Sharpless Asymmetric Dihydroxylation, Asymmetric Synthesis from 1998 to 2004. So now I welcome Boras Mandar to take over the presentation.
Sure. Thank you very much for the kind introduction, Dr. Karki and Dr. Sujit Kumar. I appreciate your efforts in bringing this webinar to the audience. And I thank all the organizing committee to invite me for this presentation. I will
Thank you,
just,
sir.
I will just share my slides. And before that, I just request all the participants to either mute them or stay on mute so that you will be able to, everybody will be able to hear my presentation clearly. And if you have any questions, either you can post at the end of the session directly, or you can share with the coordinator, Dr. Sujit
Sir,
Kumar
we'll keep
as
at the
well.
end of the hall.
Yeah, that will be
That is best.
preferred one. Yeah, perfect.
We'll take one by one.
I will just share the screen now.
Yeah.
I hope you are able to see my screen.
Yes, sir.
Yes, sir. Yeah.
Yes, perfect. What I will do is that I will go through the slide deck first, and then we will shift to the live tool where you can actually see the Reaxys, how it works or performs directly online.
That is fine.
Yeah, perfect. So before I go into my presentation, I just want to inform all the audience here that Elsewhere has opened up their plethora of journals related to COVID-19 free. You can go and access those information related to COVID-19 through this link, or you can directly go to the Elsewhere website and then visit the Novel Corona Information Center. So this is for your information there. I hope many people might not be knowing what exactly Elsewhere is doing right now. But people certainly will know us as a publisher. So we are there since 140 years plus with the publishing experience, where we have a global presence in over 180 plus countries with over 30,000 plus employees. But over the period of time, we have
Hello.
transformed ourselves
Hello.
from a publisher to a data analytics company, and we have global spends around.
Hello.
Excuse me, if you can stay on mute, it will be helping everybody. So we are spending around $1.5 billion on those technology advances where we utilize our information coming out of our journal space and then curate those. So in your next slide, you see that whatever the information we gather through our journal space, we started curating them, and then we have harmonized those content in a data where we can present it as an analytical ready tool for the researchers. Doing that, the researchers get advantage and can save a lot of time and energy when conducting the research, and we will see how we do that practically as well. So what Elsewhere has to offer to the science community, or in terms of the science and technology? You all know we have in research management, we have Scopus. You might have heard about it. So apart from Scopus, we have Scival and Mendeley as our research management tool. we also have ebook sections and journals. So we are one of the largest publishers under Science Direct there, and Cell are our Cell, Tetrahedron in chemistry are our and Bioorganic Medical Chemistry are our journals, which are useful to the life sciences people. In engineering solutions, we have various solutions like Novel, Geophysics, and Engineering Village. And under life science solutions, we have Pathway Studio, which actually gives you information, and you can integrate your live experience in terms of your target finding and target validation. I will talk in detail today about Reaxys and Reaxys medicinal chemistry, as it will be useful to the audience what we have today, particularly from the pharma background. Embase is a biomedical database tool where you get the information on adverse drug reactions and many more, particularly when the drug is launched in the clinic, or you want to modify those aspects which are actually troublesome. Uh, so you get this information, and the tool is being also used by FDA. Pharmapendium is a tool where all the data package from a compound which gets reported to a FDA or EMA, from its preclinical to clinical data, everything you will find in Pharmapendium. So if you want to modify pharmacokinetic parameters or metabolism issues or transporter issues, you can actually do those by with the help of Pharmapendium. COSA is more related to the pharmacovigilance tool. When the drug gets launched post-market, nowadays a lot of companies, since last 10-15 years, company do spend a lot of money in understanding what goes actually in the market when the drug is launched there, and what kind of adverse drug reactions or what kind of population are facing the issues with that particular drug. Everything gets reported in the COSA there. So the companies such as Infosys, Accenture, or many other software companies which are working in health, which have their healthcare division, utilize our tools to in this pharmacovigilance space there. So today we'll talk in details about Reaxys later on. So what Reaxys is all about? So when I started my PhD way back in 2000, we neither had SciFinder. I hope everybody knows about this tool SciFinder, and neither we had Reaxys in India particularly. So we had Beilstein at that time. So Beilstein is an organic chemistry database from Germany, and Gemlin is an inorganic and organic metallic chemistry database from Germany. So what Elsevier did is that they went ahead and procured these two databases. We had our core content coming from our journal space as a publisher, and we have embedded into the uh, patent space as well. So we have patent from US, European, WIPO, and since last 3 years, since 2015, we have added 4 Asian patent houses, which are particularly Chinese, Japanese, Korean, and Taiwanese, which you can see at the bottom there. To emphasize a point about this Asian patent houses is that whatever the information is required as a chemist or as a scientist, particularly in terms of the claims, abstract, or the procedure, reaction procedures, you will get that information translated into English directly in Reaxys. So one doesn't have to find a translator at least to start a reaction and understand what kind of compounds are claimed or what are the various claims captured in those patents. So you will get completely the English translated information from these Asian patent houses. So all this together brought the formation of Reaxys, which actually helped the researchers to give the shortest path to their research answer and save a lot of time, energy, and money. Now we will understand what the content is there in Reaxys. So Reaxys has a coverage of around 16,000 journals. So we don't limit ourselves to just Elsevier journals, but we have journals coming, the data coming from other journals from other publishers as well. And apart from those, 7 patent houses. So all this altogether gives you around 56 million records plus there. We do cover the substance database and chemical reaction database as our competitors, but at the same time, our property database is one of the highlight point, where we have greater than 500 million experimental properties under 400 fields and subject 130 plus subject area. So this experimental data, whatever we cover in Reaxys, is more than 10 times compared to our all competitors there. Since 2015 plus, we have also added into Reaxys the bioactive database, which captures around 35 million experimental bioactive data points, and target databases, which actually captures around greater than 13,000 targets as well. If you talk, if you see what exactly the content is there in Reaxys, apart from this overall generic information about journals and the other information what you saw in the slide, we do capture chemical and biosafety information from PubChem, and commercial sourcing information from eMolecules, Lab Network, Outreach, and many more. Uh, we also have integrated Science Direct and Scopus within the Reaxys, where you can get the experimental information directly into reaction from Science Direct. Certainly, we don't give you the access to the full text there uh, because this is a separate platform there. But you will certainly get the abstracted information in terms of reactions, substances, and chemical reaction procedures directly into Reaxys from those journals. If you are in terms of biology, you will get the biological assays. If you are in pharmacology or pharmacokinetics, you will get all those related information, and I will show them how you can decipher those information in Reaxys. From Scopus, you will get citations directly into Reaxys. In terms of drug terms, we have embedded Embase and Medline database to provide you the information on drugs. And when it comes to engineering database, particularly towards the material science and nanoscience technology, you will get those from Compendex there. When you talk about the medicinal chemistry database, what I was telling about bioactin and target, when you talk about those specifics there, you will get the information on is the compound druggable or not under Lipinski rule of 5. You will get the experimental data information on in vitro and cell-based assays, animal models, pharmacokinetics and ADME properties, and also toxicity there. So this is wealth of information for the institute like yours there. Which you can utilize them to modify your research and shorten your time space to get your research to the publication level, or a project if you want to lead that project and collaborate with an industry to get a molecule there in the clinical space as well. So particularly what happens, I'm not going in details with the drug discovery space, what exactly happens there, and we can have a separate presentation for that if you all wish to have. But just to highlight the point that main things happens is that at the starting point, a disease and a target and a compound is identified. You need to validate the target with your assays. And then after that, medicinal chemist and pharmacokinetic people come into picture, where you need to, and computational chemists also. So whatever the compounds you have hits, you modify them to leads, and that leads you then test it for safety, toxicology, and move ahead in this cycle there. You feed the information, and repeatedly you can get those ideas and modify your compounds to ultimately get a clinical candidate there. So what we have done is that how I'm showing on this slide is that how a customer, and this information is again procured from the customer directly there, how they have uh, taken advantage of Reaxys and Reaxys medicinal chemistry data to progress those steps in the drug discovery cycle there. So from their target-based screening, if you see, they have got this spiroxyindole scaffold there, where they modify that lead either to optimize the potency as Mi63, or to modify its oral bioavailability in terms of Mi219. And also at the same time, they made it more stable isomer there. So they have improved the metabolic stability of that compound. And then they further went ahead and optimized the potency there and the pharmacokinetic properties in terms of the Cmax, AOC, and T-half there. And further, they were able to achieve the clinical date. So the Reaxys and Reaxys Medchem, the wealth of data help them actually speed up this process there, and help them to modify those fragments to help them modify the properties and to get the clinical candidate. So what kind of modifications are possible with the help of the data in Reaxys? So if you see, we can get the data on ADME and tox there, as I told you. In clear terms, you also get the information on CYP450, because there's one of the main enzymes in the liver which metabolizes the compound there, right? So you get information on hERG inhibition there, which actually have the QT prolongation information there and create the cardiotoxicity. One can get Ames test, particularly this is for the anilines which actually are mutagenic there, and you get that information in Reaxys. Apart from that, you do get information for activity, efficacy, and cellular activity as well. If you or compound have off-target activity, you will get those information in Reaxys. As a pharmaceutical chemist, or a medicinal chemist, you also need information on how to make the compounds and what kind of characterization you need to do. And if there are similar compounds, you can see with the characterization data. So you will certainly get information on NMR, HPLC, UV, IR, and I will showcase those individually when I take you through the actual tool live. You also get the retrosynthetic pathways where you can get help to make your compounds or synthesize your compounds in the lab. With the help of Marcus search, you actually can understand how is your compound novel, patentable, and is there freedom to operate. So all this information you will certainly get into Reaxys. And I have went through this completely. And in terms of Lipinski rule of 5, if your compound is druggable or not, if you see here on my right hand side in the second box, there is a blue triangle there which belongs to the Lipinski rule of 5, and the orange triangle belongs to the compound. So whenever the orange space violates the blue space, you will see which properties you need to modify in terms of making your compound druggable there. So you can understand through that. And the actual data space I have captured there, what in terms of the analytical data or the target data you will get there. If you talk about the properties, if you see on the first 3 columns, these properties are mostly related to the chemistry properties of a compound. And on the right hand side, the last column, these are the properties related to a medicinal chemistry of this compound. And we'll see in the Reaxys. So everything, all this information, you will certainly get into Reaxys, and I will help you to understand how you can actually take the advantage to provide answers for the questions you are seeking out to it. We also are, in my first slide, I showed that we are partnering with major pharma industries as well as universities. So in the next few slides, you will understand what these partnerships all together, and we are also bringing in more predictive information into Reaxys as well, apart from the experimental information. So again, coming back to this drug design cycle there, where design, test, make, hypothesis, you need one of the important factor we are actually bringing in Reaxys is the match molecular pair analysis prediction thing. So we are working with the Swiss Institute of Bioinformatics there. We have our own data coming out of Reaxys and Reaxys Medchem. We have collaborated with them for their data coming out of the Swiss Institute of Bioinformatics. And this will help you to understand if you want to replace a particular scaffold or a particular part of the molecule with another another scaffold, which will help you either to improve your activity, efficacy, or the off-target activity, or even the pharmacokinetic properties in terms of the metabolism stability of those compounds, or addressing the CYP issues. So this will help you to identify what exact replacement you are seeking out of it. And if you see in this slide, there are around 95 lakh molecular replacements possible out of this effect. And this will certainly get integrated into Reaxys sooner for the prediction capabilities of the Reaxys to get improved there. Now, the next important thing what we are looking at in terms of the prediction capabilities is optimizing the pharmacology of a compound. We all know that when the compounds are similar, having a similar structure, or they are quite similar to each other, they the thought process is that they will certainly act on the similar kind of target and have produced the activity on those kind of targets. So on these similar lines, again, we have collaborated with this Institute of Bioinformatics to understand the off-target activities coming out of it. So the data from Reaxys and Swiss Institute of Bioinformatics will come together, and then when you actually put a search into it, it will for unknown target, it will dive down to the tool via the machine learning and artificial intelligence there, with all the targets and the molecules there, and then it will provide you to understand what are this molecule can hit possible targets there. And I will show you actually in the Reaxys how you can actually even take this advantage to establish a structure activity relationship as well. Now, after once you have the predictive capabilities in terms of the match molecular pairs of optimizing pharmacology, you certainly need a chemist or the pharmaceutical chemist or a medicinal chemist to make the compound actually physically, and then to see the activity of the compound if the hypothesis stays true. So for that, we already have capabilities within Reaxys to help you come up with a synthetic plan for a compound which is known in the literature, at least with a one procedure for the penetrable step. But if you have a altogether novel idea, and if you want to make that compound, and if you want to make it quickly, rather than applying your own retrosynthetic skills, we have come up with this retrosynthetic tool where actually it will help you to predict the synthetic routes based on whatever the starting materials available in the either the commercial space or the intermediates with you. So we have collaborated with Waller's lab, and there is an article in the Nature there from Professor Waller where we have used this artificial intelligence and machine learning technologies to come up with the synthetic procedure or synthetic route, particularly for a particular unknown compound, whatever it comes to your mind to when you have designed that compound. To address a particular activity, efficacy, or properties related to pharmacokinetic things. So this tool actually is 30 times faster than the actual traditional computer-aided methods available in the market there. Also, it is almost twice, it actually provides you twice as many solutions in terms of the molecules there, right? So this is what we have as a separate tool. Right now it is available to the industry, and it will be launched for the academia in the next year, early 2021. So that the academia, if they want to have this tool, they can have it from elsewhere. When in terms of the collaboration, which I was talking about, I have a couple of examples where we have collaborated with the academic groups. And this is one of the example from Jensen's research group, where the content from Reaxys and Reaxys Medchem has helped him to come up with around 12.5 million single step reactions tabulated in the Reaxys database. From this, he was able to come up with more than 1,63,000 compounds in terms of libraries. When we talk about one of the collaborations with the Lapkins groups there, right? So he has utilized the information from Reaxys Medchem to find out around 516 molecules, which actually will might drive the waste-free economy there. Apart from this academic collaboration, we also have collaborated with various pharmaceutical companies. So AstraZeneca right now, this is an example where we have our own content coming out of Reaxys and Reaxys Medchem. There is a patent space data available there in the patent space, and the AstraZeneca had their own content in terms of activity information, in terms of synthesis information. So what we have done is that we have helped them integrate everything in single platform in within Reaxys there, and which actually now comes up around more than 20 million reaction space when it is a integrated content there. And this have helped them to speed up the design, make, test cycle there, and help them come up with novel compounds, which or the new chemical entities which might be a possible drugs there. Apart from AstraZeneca, we also have collaborated with Bushi Aptec, one of the largest CRO in the world, or Merck, Roche, Lundbeck, and recently now Novartis is one of our collaborators where they are completely on the Reaxys platform there. So what in terms of the content there? So whenever articles get published in journal space, it takes around 11 days to 15 days to get that completely accepted information into Reaxys. And when it is a patent space there, it takes around 30 days to get complete information from the patent into Reaxys searchable form there. So in general, we have a platform which actually addresses from life sciences space within the target identification, target validation space, to how to make the compounds, how to identify the compounds, how to improve the pharmaceutical properties of a compound. And then it also provides you information of safety, toxicity, where you can get the information on the no observed adverse effect levels to what will be the minimum efficacious dose or the maximum tolerated dose within Reaxys space. And from the content what we have greater than 500 million experimental data points, which will help you or enable you to drive those decisions. Now, how it will be useful for a researcher or academia? When you talk about academia in terms of the chemistry or medicinal chemistry space, you need to have a novel idea generation, right? So you need a tool where it will also help you to generate your proposal or grant right up there, right? So Reaxys helps you with the wealth of information in coming up with the ideas and helping you understand a particular project, and help you also within the write-up of those proposals from the data what it provides there. It also helps you how you can design your compounds and are your compound novel enough or patentable enough with the help of the Marcus search there, right? And these first three boxes remain same. Either you are a chemist, medicinal chemist, a pharmacokinetic scientist, or pharmacognosy scientist. Everything is similar for them. And then when you talk about the synthesis of protocol of novel compound, Reaxys provide you all those information on the synthesis, analytical data. Then when you talk about optimizing off-target activities or toxicities, Reaxys provide you information on those as well. And then when you want to talk about translation of those activities into potencies or efficacy, how you can profile a compound in terms of the pharmacokinetics, in terms of dosing, half-life, or Cmax AUC, Reaxys do provide those information as well. Now coming to your discipline, the pharmacy disciplines. So according to my knowledge, I observed that these are the major 5 disciplines within the pharmacy there. Either it is pharmaceutical chemistry, pharmaceutical analysis, pharmacognosy, pharmacology or toxicology there, and pharmaceutics or biopharmaceutics there. So if you talk about all this discipline, Reaxys and Reaxys Medchem do have the content to address everything within your pharmacy main discipline there, right? And have listed in the green color what content we have, which will address your individual disciplines or departments there with the type of data within the Reaxys there. And as Dr. Karki said, we will take those questions at the end of the presentation, and I will now share my screen, which is the live tool Reaxys, and let me know if you are able to see my web page there of Reaxys. Anybody can confirm there. Dr. Karki, are you able to see my Reaxys page? Hello? Hello? I hope.
Yes, sir. I am seeing.
Perfect. Thanks for your confirmation there, right?
Please continue.
Yeah. Yes. So this is what Reaxys page looks like. You can type reaxys.com in any browser, be it your mobile browser or any internet browser, surfing browser on your laptop. You can actually access Reaxys. Whenever we provide access, it is based on the IP. And if you are in the Wi-Fi of the same institute, within the institute, you can access Reaxys on your mobile. You can actually log into the, you can create your own login within the Reaxys there, even though you have IP space, and this will give you more advantages there. And I will talk the advantages, whatever you see there. You can actually have your own specified user interface the way you want as well within Reaxys. You can create, you can save your searches within Reaxys when you create your login. You can create email alerts to your searches as well, and I will show you how you can do that. Then we have two basic searches. One is the text search, typing. You can type your searches, and you can draw your searches there with the searches, and I will show you the advantages what we have there. Then we have a query builder where we have greater than 500 million properties there, where you can do mix and match searches. Within the query builder, you can create more complex searches there, and we have designed forms, pre-designed forms for you, or you can design your own forms and save the forms on your space there within Reaxys or on your computer there. We have a synthesis planner which will give you a retrosynthetic scheme there, how you can make that compound there with the experimental references for each step there. As I told you, you can save your searches in the history space there, and you can create email alerts there, right? So this is how what it looks like the web page there. At the bottom of the you will see what contents are there from Reaxys there, and this gets updated quite frequently there. But because of the volume of content, like it's 118 million substances, so even though there are 10 compounds added to it, you might not see the difference there. But over a period of time, you will certainly see these moving to the new numbers there based on the content getting added there. Right. So let's go and type any letters just to see what text search you will get it, right? So I'll just type 3 letters, say Suzuki reaction. If you want to type, say, being a pharmaceutical chemist, you want to see understand what reaction is there, right? So when you type 3 letters of any search, what Reaxys does is that it will show you 10 different options. So as a researcher, what it will helps you is that even though you might be looking at a one particular question, or you want to understand a particular search query, if there are 10 different options provided in front of you, as a researcher, it gives you more power to seek information from those particular search information coming out of it as options there, right? This also helps in learning for the students as well. And if you keep going further, the reactions options keeps changing also, if you want to see that, right? So this is what the text search looks like. And when you comes to the drawing search, we have three ways of drawing there. We have provided Marvin JS as our Java based tool, drawing tool, which is much more powerful than any other tool. Also, we have provided the ChemDraw tool to the researchers within Reaxys. And again, our computer doesn't have the ChemDraw within their own platform. You need to import or the ChemDraw structures within their platforms. But Reaxys provides you with a direct tool there. The advantage of this is that you can copy paste from ChemDraw, Word, PowerPoint, or any format of those structures within to Reaxys, or you can copy the structures from Reaxys to Word, PowerPoint, ChemDraw directly there, right? And the third way of drawing is to text to search. Again, this is very unique features, and this is one of the best features. It will help you when you try to access your Reaxys on your mobile, because it's very difficult to draw the structures, and you can just type a name of a compound there, say dexamethasone, which is again a drug repurposed right now with the Cadila. It is available for COVID-19, right? If you want to, you just type that compound, and you can get the structure directly into Reaxys, right? And it is not just the compound in itself. You can have a generic name, a IUPAC name, a common name. Say you want to have a dipeptide or a tripeptide, right? Or a decapeptide, right? It will directly come and draw those structures for you. And then you can edit those structures directly, right? So with the set based amino acids, you will get those structures directly. Or you have, say, oxidizoles or IUPAC name. If you type it, you will directly get those compounds directly into Reaxys. So this is one of the best advantages of Reaxys, where you can type a chemical name and you will get it there. On the right hand side, if you see, there is a related Marcus search where I'm putting my mouse pointer right now. And this will help you to understand if your compound is novel, if it is reported or not. You can do those structures. You can do exact searches as drawn. You can have a substructure search or a similar search, right? And I will let's take you through the more powerful drawing examples also. I will spend some 5 minutes here, and then we'll go further, wherein I will address all the pharmacy disciplines based on the content we have in the Reaxys. So the advantage of the drawing tool is that if you go here, we have a periodic table here now. And if you see, there is an atom in the list and atom not in the list. So you can ask Reaxys to search if you want, say, in the search pattern nitrogen. Sorry, I will first select atom in the list. So if you select, say, nitrogen, oxygen, chlorine, bromine, um, and say okay, and use this. So now Reaxys will search, uh, all the searches will contain only these elements, like the amino group, nitro group, or the oxygen with the hydroxy group, that is phenol, chlorine, bromine. So you will have those only elements coming in their searches. So when I transfer the query and say find, uh, you will see those substances. Now, the advantage again here, if you see in the search result, is that Reaxys segregates their searches, and based on the need of the user, you can go there. So if you are looking exactly the way we have drawn, so you will have the substances there as 6,800 substances. If you are looking at the information or targets where these compounds are active against, so you can get the information on targets there. If you are looking at the average similarity of the substances, you can choose those information. Or if you are directly looking at the reactions of from these substances, you can do that. Whereas our computer gives you a very cluttered information, and there will be multiple hits coming out of it. And now when you preview results here, you will see that whatever we made the searches, it will have those functionalities coming up in our searches only, right? It will not have any other thing. And now if you don't want these particular choices, we can go back there and we can change our search pattern there. So say edit search. So nitrogen, oxygen, chlorine, bromine, we will now say atom not in the list. So this our search will not have those. So this is one of the unique feature of Reaxys. So say not in the list, and then say nitrogen, oxygen, chlorine, bromine, and say okay. And now Reaxys will not show you these results where you have these elements or functional groups as your search hits. Right? So you will have other functional groups coming up there. So this is one of the biggest advantage. Now, what is the other advantage? Now you talk about say toluene itself. Let's take this example here. What data you will get it? As I was telling you, it will see based on Lipinski rule of 5, your compound is drug-like or not. Yes, it is very small compound. So certainly it will be fitting within the Lipinski rule of 5, but you will get that information there. And we will see for other compounds as well. You will also come to know the identification data. So chemical name, it is also methyl benzene and toluene. So you will get that information there. You will get various forms of Inchi there. You can copy paste this and come out with the structure there. What type of substance is there, you will get that information there. In the other data, you will get information on how to handle this particular reagent, say exposure assessment, right? So how much issues are there with the exposure of this compound, what kind of issues you will get in there. So you can go and read these articles directly, and you will know the information on how to handle a reagent through this information there. You can actually understand the biodegradation, bioaccumulation data, transport, and this, any related data to that compound. You will also have information on say physical data points, how to address this, either melting point, boiling point, refractive density, and there are many more there. I am not loading. So there are multiple data points there. And as I told you, we have greater than 500 million experimental properties, and this is how you get. And we'll see those data with another searches as well. So, but if you want, I can show you say density. So you will see the density, you can see the measurement temperature at which it has been taken, what is crystalline, and you will get that where it is located in that particular patent or a space there, right? If it is supporting information, you will get into the supporting information location, and then you will get actual reference there, right? If you are looking at say spectral data information, I will just close this and we'll see the spectral data information. See, melting point is there, you will get the information on the melting point directly as a tabular form, right? There are 7 hits here, so you will directly get that information on the melting point there and their references. Now, if you want to see a spectral data of a compound, say you want to see IR of this compound, toluene, where it will go, and you will come to know the information on infrared spectroscopy, right? What was the solvent or the spectroscopy method, what are the bands, and for the bands of the spectrum, you will get that information directly into the references there. You can go and see those references there. If they have written directly in the experimental section, we can actually get direct written here itself, and you will see that as well in this one. If you're looking at the mass of a compound, you can go and see which type of mass spectroscopic analysis they have taken and where it is located in the patent or space there. You are looking at absorption spectrum, you will get an information of absorption spectrum, in particular UV visible spectrum, and what solvent and everything is there. If you are talking about the NMR spectroscopic information, you will get that information directly in their space there, right? What solvent, what temperature, if it is taken a temperature dependent NMR, what kind of, is it a 1H NMR, 13C NMR, a DEPT, you will get that information directly there within Reaxys. So what it shows is that in a particular compound, all the information related to the compound is available directly there and there itself, right? If you are talking about the bioactive data points, you will come to know for toluene particularly, if it has a efficacy data, in vitro data, activity data. Again, it is a tabular form, right? 32 test results with various literature references, right? Again, you will have the actual activity. We have converted this activity into a px number. It is a logarithmic scale because in various literature forms, the activity gets reported in terms of the micromolar, millimolar, PIC 50s, right? So to compare head to head, we have converted that into a px value, which will enable you very nice and direct comparison with each other, one-to-one comparisons there. But you will certainly get the actual numbers as well and a reference to that articles directly. Similarly, if you have any toxicology or safety data, you will get that information on that compound directly there. Animal model data. I'm surprised to see even toluene has animal model data there, right? Directly there, right? And what was the route of administration and everything is captured there directly in this one. So if you want to see preparations of the toluene, you can see you can see how toluene can be prepared directly. And I'm taking just an example there just to showcase the power of this tool, right? So you will get directly the experiment procedure in the Reaxys itself. You will see here, you can directly see those information directly in the Reaxys. You do not have to go and find out and read whole patent to find out the procedure for that particular synthesis of a compound. And if you, out of this 3000 odd references, say you 3527 reaction, you just want to see the results where the Reaxys have captured the experimental information within Reaxys. What you can do is that you can go down on the left hand side filter panel here, and you can click the box experimental procedures only, and you say limit to, and Reaxys will now showcase those results where we have captured the experimental procedures directly within Reaxys itself. So you just click on the experimental procedure, you can get that information directly. You have yields information also. Now, out of these 349 reactions, you researcher want to see those reactions only where the yields are greater than 90 or 95%. You can directly go to the again filter panel there, and you can filter those results direct, right? Say select 90 and 95 above and limit to. Now you will have from toluene preparations, you have now uh, the data coming out of the experimental procedures captured in the Reaxys, and from there we went to showcase the best procedures depicting greater than 90% yield, right? So this is how you will get those. If you want to again, you can go back to the maximum results there. And if you want to see where toluene is used as a reactant or used in the reactions, you can directly go and see this information where toluene is used as a in reactions there, right? So it may be a solvent or anything. So this is how the reactions of toluenes are there. It may be preparations or it can be used as a reagent as well, right? Let's do another search now to understand from structure what we see that, right? Uh, So now if you want to create more R groups or a defined R groups, what we can do is that we can select this particular hexagon cyclohexane ring with a bond inside, and you can select like this. What it does is that now you can, these are the substitution patterns for you. And let's say this is a say aniline there, right? Now you can create R groups same way by selecting atom in the list, atom not in the list, or individually you can select those, or you can directly create a functional groups there in terms of smart R. So if you select this R here as a smart R group and click here, and if you want to rename it, also double click this on here, and you can select R1, R2, whatever you want to do that. And then now you can create the functional groups directly there. Say you want to have a say alcohol there, right? Secondary alcohol. So secondary alcohol, and then you create this branch there. You want to have acid group there, say. So you can create acid, or you want to have a isopropyl group or a methoxy group, right? So you can create that as well. So you can create methoxy, say, right? And now you can group this, select again the smart R, and then you can group this. It will create your R. So what it has done is that you can actually specify what functional groups you want on a reagent also. And this is very unique to Reaxys. Again, our computer doesn't offer this kind of depth of drawing power tools. And now your substances will have uh, the results will have those functional groups what we have drawn or their substitutions, right? So you will see those coming out of it, and it will be at the ortho meta para because we have selected everything on that one, right? 2, 3, 4, 5, 6, everything is there. So we will have those coming up in terms of the aniline there, right? So this is the power of the drawing tool. I am not going now in more details about drawing because this is just to showcase how you can do that. Now let's take an example of a drug or the active. Let's say take the example of the same dexamethasone itself and see if we can find a procedure to make that compound, right? Say transfer query and say find, and we are doing directly as drawn exactly. So now it has shown 858 results for dexamethasone. And why? Because they might be salts, mixture, isotopes, charges, radicals, right? So because of that you see those, but it
Hello.
will certainly be a dexamethasone.
Okay.
You can directly, if you want information on where the dexamethasone is hitting the targets, you will directly get those. So say view results. So now if you see, this is the information of dexamethasone, and now you want to see how you can make that compound, right? This is a very complicated compound and you need to see the synthetic procedures to make this compound step by step. So you can go and ask Reaxys to create a synthetic plan. Either you can do manually one by one, or you can ask Reaxys to make auto plan for you. Reaxys can help you to make 10 different synthetic route procedures for you at a time. You can actually have a linear or convergent synthesis designed for you. You can choose maximum 10 step in a particular synthetic route. If you say yes here in terms of the starting material searching, which is commercial, Reaxys can stop the searching immediately when, even if the starting material is large enough or costly enough. But if you want a smallest possible or a cheapest starting material, you can say no, and you can ask Reaxys to search synthetic procedures for you, or synthetic route rather. And you can modulate the yield of a reaction as well, whatever the overall yield of a reaction. And then you can ask Reaxys to create plans for finding what kind of synthetic routes are possible to make dexamethasone itself. Again, as I am using my 4G connection, a little bit speed issue might come here, but certainly you will get the synthetic routes to make dexamethasone here. Right. So here you go. So here is synthetic complete protocol, how you can come up with the dexamethasone molecule in itself, right? And every step you will have conditions there. So you can say show conditions, and if you want to see at the top, here you go, the experiment procedures captured in Reaxys. Every step you will have, whenever you click on this 3 dotted line, and you can say show conditions, you will certainly get those protocols there, right, directly. And because I have selected 5 different plans, you can go and see various 5 different plans. See how small or various groups it can go there, right? So this is how you can make the dexamethasone there, right? Let's go back to the results again. Right? You want to see information on the targets now. Say this is what compound is there, but I want to know which kind of various targets dexamethasone is hitting. On the right hand side, you will see the most active compound for that particular target. So this is the cyclin adenosine monophosphate phosphodiesterase target there. And if you want to see the more target detail, if any assay information is captured in Reaxys, you will certainly see that. You see here the assay information, it has the data, what cell lines is there, and what is the activity range, right? You will get that information directly there. If you want to see the protein information, you can directly go to the UniProt site there, if it is captured there, and you see substance and document information for those particular targets captured here directly. Let's go. And now if you want to plot dexamethasone again, all the targets there, you can do directly like that. So you say heat map, and you can observe the SAR based on this one. So you can plot targets or target species, biological species effect. Multiple parameters are offered to you to plot the graph, and then you can have substances, and you can apply that to see dexamethasone as substance against various targets, and you will see the list of the activities there captured. Right? You can do that way also. Let's go to the results directly there. And then I already showed you in terms of the bioactivity data, what it provides you in the information there. Everything is captured there. If you are looking at metabolism data, metabolism is captured in there. If you are looking at the pharmacokinetic data, I was telling you in terms of Cmax, AUC, what animal model the pharmacokinetic assay has been done, what is the route of administration, everything, and what is the dose. All these parameters are directly captured within the Reaxys for that particular compound. Right. Now let's take, I'm switching gears and I'm trying to take you to the more complex queries. Right. So now this is what the query builder is look like. And if you see on the right hand side, we have created individual forms directly there. So rather individual properties there. So you can search by topic keywords, you can search by identification of a compound, be it a chemical name, element, symbol, all this individual properties you can select there directly, right? So much information is there. Then you can go to physical properties tab if you want to select a compound or a physical properties and you want to add that. You have to just click on it and it will populate here and you can create those, right? Um, we have Reaxys properties as a chemical properties, and we have medchem properties also. So if you go individually on the medicinal chemistry properties, we have substance action on target, substance effect, measurement of px. So action of target can be an inhibitor or a blocker or antagonist, agonist. All these actions are if you don't know, you can directly go one by one and you can look at those individual also, and you can have. We have then information on the route of pharmacokinetic people. You have information on route of administration, dosing regimen, what is the dose. Based on the dose searches also you can do those. Based on if it is in the clinical phase, which clinical phase, you can do those searches also. If you hold on a target and you want to understand a particular assays, you have these categories in terms of the bioassay categories or bioassay subcategories. Name, animal models, all these individual parameters, you can do those searches. If you don't want to do that, individual parameters searches, we have created a pre-designed forms for you in terms of Reaxys as well as Reaxys Medchem forms. So you can utilize one of those forms. So if you want to say, I want to understand the cytotoxicity of a compound or hERG inhibition issues of the compound, you can click directly and we have created that form, right? And then you can modulate, you can add a structure to it. You can change this. If you know, hERG is a potassium ion channel, voltage gated channel, right? So you can say if it is a wild type or a human or a mice potassium ion channel, you are looking at it, you can do that. You can change this measurement parameter as well there as well. And you can utilize these Boolean operators to add this and not, near proximity. And I am not going to details. We can have a separate presentation if you happen to subscribe to that for the training as well. So you can understand this off target or liabilities of within a compound. I was telling you about CYP data. You have this information of CYP. Same way, microsomal stability, protein binding, all those parameters in terms of pharmaceutical properties, you can actually do those searching as well, right? So this is related to the pharmacology of a compound. Now, if you are in the field of pharmacognosy and you want to understand the isolation of a compounds, right? So you can say isolated from a natural product, say, source, and you can actually search based on molecular formula or molecular weight also. Say molecular formula will do that first, and say you are looking at C25 and above. So carbon 25 and the other elements will be captured when you put a star there. And now you want to add a property to it, right? Say you want, say, anti-cancer property, say, suppose cancer, and we'll put star, so that everything after anti-cancer also will be captured. So what we are now creating a search is we are trying to search isolate compounds isolated from natural source, or they are natural products with carbon 25 or more chain length, with other elements captured after carbon, and it will have anti-cancer properties, right? And then you hit substances, say. So this is the level of complex searches you can create as well. So now Reaxys has shown you the data of 24,000 odd compounds which have anti-cancer properties, and they have carbon C25 or above, and they are isolated from natural products, right? Now, if you want to understand which are clinical compounds, which are in which phase or market products, you can, or you want to sort them based on that information, you can go there on the left-hand panel, and you can say, I want to understand only marketed compound there, or which are in phase 3 or phase 2 compounds, right? So you can select those compounds and you can limit those searches directly. You want to see which are the most active compounds. So you can say select px7 and above, and you will see say we will select 9 and above to limit our searches, and you will now see the compound which are the most active out of this lot, out of this 24,000 odd compounds which are showing anti-cancer properties and isolated from natural products. You will have now 584 compounds which are the most active out of those, right? Now, if you want to altogether study these 584 compounds against their targets, you can again go to the heat map, create a structure activity relationship for you, where you can plot substances against the targets, and you can plot those graph which I was showing you earlier as a heat map there. Now, on the columns section above, these are all targets where these 584 compounds are active, and on the left hand panel down, which are in the rows, there are structures of these compounds, right? And now, if you want to, suppose you have a hold on to a particular protein or a receptor there, and you are aware of the information on that particular receptor, or you're working on that particular target there, right? So what you can do is that you can say select that, and you can sort activity based on that particular receptor. So now the left hand side compounds, the rows will get rearranged, where the top one will show the most active compounds on this particular targets and its activities there. And now if you want to see information of this particular compound, what is this? So you can go to the all substance details, and it will show you again every information related to this particular compound, right? We have hit data of that compound, we have identification data, we have drug likeness data. So everything is captured data. Patent specific information is there. You want to understand now drug likeness of this compound. This is a large molecule, but you want to see if it is fitting in which properties. Now, if you see here, hydrogen bond acceptors, you need to modify it because they are 13 and you should be below 10, right? Donors should be below 5, so donors are less here. But which property of a compound in terms of Lipinski rule of 5 you need to modify, you can actually understand from here directly there, right? You want to understand spectral information, physical data, identification data, you will come to know. If you want to synthesize the compound I showed you, you can go to here and autoplan and see how you can make this compound. So this is how you can do that. Let's go to this 584 substance again. Now we have selected this based on a particular protein, but you want to see apart from this, you can lock this also. So you can say limit to selected columns. So once you do that, what will happen is that because you are having this protein in hand or information on the, or you are working on this particular protein, now you want to see, forget about this 584 compound, but whatever the compounds active against this on this target, you want to understand, you can directly go here and say limit to selected columns. Now the columns will get rearranged, the rows will get rearranged, the substances will get rearranged, and you see now there are only 7 substances out of 584 substances which are active against adenosine A2A receptor out of the so these 7 substances have anti-cancer properties and are isolated from natural product. So based on our earlier searches, this is narrowed down like this. So this is how you get the information, where you can zero down to a particular searches, you can understand what compounds are there, and you can modulate those things. If you want to understand the target details, you will come to know directly from here, right? So this is how you can modulate. Now, similarly, you can again go back to those 584 substances in the SAR. And if you want to understand based on a structure there, right, heat map, you can do that also. Similar way, we limited ourselves to the column. We can also limit our sort based on the structure itself. So now we have plotting all 24,000 odd substances against all the targets there. And now you can actually sort based on here as well. See, exclude or limit to the column, right? You can exclude this row or even see. So based on this compound, now if you limit it, you will see which this compound active against which possible targets. All those targets will be lined up against, right? Straight away. So this is only an enzyme, a particular enzyme, and you can, if you want to know the target details, you will come to know there directly. So this is how you can play around with the SAR directly there. You can modulate, you can then come up with a synthetic route itself. And if you want to play around within the SAR table, you can select the filter panel here. If you want to know the data only on the human targets, not the rodents or the animal models, right? So you can you can select those target types, and then you can actually select the target species there, and you can say humans only. So you will have the data only on humans, or if you want to have rat, mice, molecular, you can do that. If you want to play around log P's of the compound, if you want to modulate those, and if you want to understand, you can filter those informations based on the log P's as well, right? Uh, if you want to search which are the latest compounds or the latest compounds out of this lot reported here, so you can type, you can modulate which is reported in 2020 or latest. There are 5,000 odd compounds which are published in 2020 or so. So you can limit based on the publication year, document type, all this information you can do that, right? So here you go. So these are the compounds which are reported in 2020 there, right? So we'll exit this full screen there. We'll go to the substances back. Right. So let me take you to the history now. So we have done so many searches right now. And if you see, you can, as I told you, you can save your searches directly and you can create alerts. Similar way, you can, even if you go to the results also, we have given options here itself directly. See, if you take your cursor here, you can create alert directly here itself, and you can then you can set the frequency of your alerts as well. Let's go to the creating alerts thing. So if you want to create alert for this particular search result, you can name that thing, what frequency you want, every week to check on it, or every day, or after every update you want information on that particular search result, you will get that. If you want after any update, or only after one first appearance in the database you want to update, you can create that information there. You can, what exactly you want in the email, you want complete abstract, full abstract, partial abstract, or only hit details, you can choose those options for your alerts as well. And Reaxys will shoot you an email if any new information is available or added to the Reaxys for that particular type of searches you have created, right? Now again, go back to the query builder. Let's say sorry, if you are looking at a particular property, say you are looking at a isolation of a compound, say HPLC, you are looking at it. When you are doing in a pharmaceutical analysis, and you want information on, say, HPLC, and say you are looking at a Taxol as a compound, right? Say, suppose, and you hit Reaxys. Reaxys directly will show you the information on the HPLC of a compound, particular compound you are looking at it. You can do either by structure search, or you can type it. And if you see hit data, there are 6 hit data with the chromatography data information there. So out of this 13, there are HPLC information for this, and you will have direct references coming up there. See, right? This is 10. Here we are captured at what minute. Also, it is isolated there. So directly into Reaxys, you will get this information. You do not have to go read the particular article and do those searches there. Even the thin layer chromatography information will be there. If you, instead of HPLC, if you want to type TLC, say thin layer chromatography, right? And of the Taxol, let's see if you have that information there the same way. Yes, I think there is one result directly for Taxol with the TLC information there, right? And here you go, TLC chromatography information there. RF value, it is captured here exactly, right? So if you want to look at a particular substance property information, you can even do those text searches directly, and you can find that information there. Also, you can take from here, you can go edit in the query builder search, and you will understand how the search is designed. So if you see how Reaxys reads that, it has, the Taxol has been read as the structure itself directly, and this is the chromatography data information as a field directly there, where you can say TLC information, which is original string and everything. Again, you can use Boolean operators. You can change this, and you can modify those changes to it. You can add any other property information, right? Say you wanted, instead of chromatographic data, HPLC data, now you also add HPLC information there. Sorry. it will be again in the chromatography information. So you can add again one more field of chromatography information, and now you can add fields to it. What kind of chromatography data you want? So select. This is everything related to chromatography information captured here completely, right? You can create those purification SFC information. So as a pharmaceutical analytic people also can utilize this information for compounds. You can do substructure search and do those information capture it here. So it is for pharmacokinetics, pharmacology, pharmaceutics, because we have metabolism information there coming up there. You can take that information and modulate those. We don't in the chemistry section you can have the information on how to prepare your aesthetics as well, reagents. And then also the pharmaceutical chemistry, we captured everything, the information related to pharmaceutical chemistry and pharmacognosy as well. So I was able to showcase for institute like yours how this Reaxys tool will be useful for not only the learning for your research activities as well, be it a student or a researcher or a faculty there, and it captures each and every departments within the pharmacy discipline itself directly right there. I think we have now 15 minutes left for our webinar, so I will be happy to take questions, and we can extend for 5 to 10 minutes also if there are more questions also, rather than showing complete details about the product. And if you happen to have subscribe to Reaxys further, and we can set up a separate training sessions for your institute directly, wherein I will be able to guide you or showcase you how you can still utilize to get more out of this product. I hope I was able to address everything in your mind in my presentation, but if not, then we'll certainly be open for the questions. Dr. Karki.
Yeah, I'm just happy with your presentation, and it was I'm getting so many messages,
Okay.
and because it's not ordinary webinar, the most of the webinars attendees are very happy with your presentation. So now we'll open for the questions. Hello.
Yes, sir.
Yes. Please,
No.
Dr. Sujit.
Yeah, I request all participants to please raise your hand if you have any query or any questions, so that it can be answered. Okay, we have one question from Mr. Nishant Joshi. I
Okay.
request Mr. Nishant Joshi to please unmute yourself and ask the question. Mr. Nishant Joshi, yeah, please ask question, sir.
Good afternoon, sir. This was a very nice talk. And
Yes.
I am a PhD student. I'm working on the Plasmodium falciparum. My PhD
Okay.
was on Plasmodium falciparum.
Okay.
So I want to test different molecules against a protein.
Okay.
I wanted to ask. I just checked that this tool is very good, but this is not freely available. We have to pay for it. So how
Yes.
is it charged?
So that information we will discuss it with your faculties there. But certainly, if you are interested, you can convey this to your faculties. And
Okay.
as Dr. Karki also was interested, we will talk. If you are with the KLE institutions, then we will talk with the professors, and we will come up with the pricing for your institute so that you can have unlimited access based on IP.
Yeah, I am not from Delhi. Actually, I belong to the JNU Delhi.
Okay. So what we can do is that you can, if you want to conduct a similar webinar for JNU, because you are only one of the participants from JNU, then what we can do is that we can, same way we can showcase there, and then it will be, if more people are interested, we can actually talk to your professors directly and do that, because let people understand what this tool about. Is it really useful for your complete institute or a particular department, and based on that we can then set up the pricing and everything.
Okay. So my question was, this is not available at individual level.
No, it is not available at individual level. So what you can do is that I will just shift my screen sharing to my presentation earlier, and you can note down my
Details,
phone
contact.
number and email addresses.
Yeah.
Here you go, so that you can write to me directly or contact me, and then we will set up a separate session for your institute level as well,
Sure,
or
sure,
we
sure.
can
I will.
talk offline. Yeah.
Okay. Thank you.
Thank you, Nishant. Any more questions? Participants, please raise your hand, and if you have any question, please ask. Yeah, I repeat, participants, if you have any questions, please raise your hand and get it clarified, or else Professor Mr. Madas will move to the next. Okay, sir, I have one question.
Yes, sir.
Yeah, sir, actually, I am working on drug design, drug discovery part.
Yes.
So we do molecular modeling, and then we predict the targets, all the steps we follow. So I found your this Reaxys very useful. Why? Because usually we do different server based search like drug bank, PubChem, SysADME, and then we do KI So this Reaxys is like it is covering everything in one platform, right, sir?
Right, right.
So my questions are, does it predicts or it is only used to search? Like you said, the example of dexamethasone or Taxol. So if I have synthesized new molecule, design new molecule, and if I want to
New molecule.
new molecule, and if you want to know those
Targets.
parameters, properties, will it be give information like synthetic planner route, especially as a chemist, like synthetic planner? Can we get the synthetic route for my design molecule or
Yeah.
for imported molecule?
Yeah. So it's a very, very nice question. So for a completely new molecule where there is not a single report, you might not get any information particularly towards the synthesis in Reaxys. But how you can do that? You can do a substructure search. So similarity search. Before that, I show you. I just want to showcase because you mentioned as a computational chemist. If you now and I will come up to compound data here. Say I am not able to get it. But if you want to export all this compound, say information, we have information export in the terms of SD-mol file. So you can actually play around with the compounds SAR in terms of the chem informatics as well. And you can integrate that with your current compound data as well when you are a molecular modeler or you want to do as a chem informatics person. So this also features is there in Reaxys, right? When you talk about exports things.
Okay.
Now coming back to your question regarding the synthesis, say you are designing molecules based on say Taxol only. Let's take the example of Taxol or say dexamethasone, whatever you want to take. Say Taxol, I will type. Now, because your molecule be new and you are not aware, what you can do is that you can do a substructure search or a similar search. So when you do that, similar type of compounds will show, right? And then if they are known around, so if you modify say here, say fluoro or say put a methoxy here, and now you want to make the exact compound, you might not, if you say as drawn, you might not get the result. Let's see if you are able to get any results as drawn. So there are 0 substances, right? And now when we go and edit that and do a similarity search, what you will see is compounds, most likely it will showcase where they are having the tight similarity, near similarity, average similarity, right?
Okay.
And say let's take a wide similarity. See, so many options gets created, and now you will actually get the designs also in your mind. Say when you do the similarity search, right? What type of modifications you can possibly do on a compound also. And then because it's a large molecule, you might not understand the differences here in the structure itself, but if I take a smaller compound, you will be able to understand, and from there you can come up with a synthetic route or a synthetic design, because that particular compound reported will have a reaction scheme, synthetic protocol, and you can utilize the similar type of scheme in your molecule, right? Though adding that particular functionalities.
Yes, sir. Got.
So, Professor Dr. Mandar,
Yes, sir.
for new compound, I think you'll not get any targets.
But you will, yeah, for new compound, you might not get a target, but when you do substructure or similarity search on that compound, you might get a target information. Or you can do the other way around. You can do, if you are having you are working on a particular target, say you are working on COX-1 COX-2 inhibitor, or any say you are working on a kinase, you can search based on that particular target and get the compound list and then modify those compounds, right? You can do both ways.
Yeah, we have one more query from Mr. P. Bharat.
Yes.
I request Mr. Bharat to ask this question. Unmute yourself, Mr. Bharat, and ask the question. Mr. Bharat?
Bharat?
Okay, Nishant has again raised his hand. Okay, Nishant, unmute yourself and ask the question, please.
Yes, sir. So I have one more question. So,
Yes.
as far as I know, actually, I am related to biology, not in chemistry. But as far as I know, we can do QSAR studies with the compound cheminformatics. So, does this tool has embedded 3D, 2D QSAR we can perform directly with this tool, or we can get some information here and do the 3D and 2D QSAR in some other place?
Yes, so you do not have the 3D and 2D QSAR within the Reaxys. They are separate tool. But as I told you, based on the export function, you can actually export as a mole file or SD file, and then you can actually embed those information, whatever you need. And when you talk about the exports, let's run any molecule, say, let's run a very simple molecule, indole, and do a substructure search, right?
Mhm.
Because that will have more compounds coming up there. And do a substructure search on all atoms, and now you will have huge number of compounds coming up there, because it's a very small molecule we have taken.
Yeah.
And I will show you how you can export and what. See, there are so many substances coming up there, and you have the targets. Now within Reaxys, if you click on even the substances, you can toggle between the targets, documents, reactions, and directly above in the top. Now,
Okay.
if you want to export, say, if you want to, and Reaxys gives you exporting of at a time 5,000 compounds or 5,000 documents at a time. Okay, so you can create a range, because you cannot export all at a time results. So you clearly read the selected export range exceeds the 5,000 compound. So you can, what you can do is that you can put a range. Range and say 1 to 5,000. So right now I will select 1 and say 1 to say 1,000. I will put just now for sake. You can now go up to 5,000. And now what data you require, you can choose based on what your need. And then here is the options formats, what you need to export. Because you want to do a QSAR, you can select SD mole file, and then you also can select what data you require also. I think just a minute. So choose specific data and add data points. Now you can go to the data points, and as a biologist, and you want to do a QACR based on the activity or a particular property of a compound,
Yeah.
you can go to a medicinal chemistry data. You don't need a chemistry data, so you can select the medicinal chemistry data. You can go activities, drug likeness. If you don't want, you can remove that drug likeness. You can go bioactive data. Now individual assays information also, you can select what you want exactly. See, so exactly you will remove every other information which is not required for you, and you can get the exact information what is required to your model, transfer to your model, right? This is how you can play around and get the specific modeling done.
And yeah, so I have one more query.
Yes.
So in other some other tools like Swiss Target,
Mm-hmm.
there also they predict the target for a particular compound. So
Mm.
but the limitation there is they have very, I think, only Homo sapiens and mus musculus as a organism.
Okay.
And my work is on parasites. So does Reaxys have more organism in the database, or it also only has the musculus and Homo sapiens?
So this will come under which targets you are saying? This will comes under uh,
Suppose I have a compound and I want
species.
to
You are
find,
looking you are looking at a species, right? These are species.
yeah, these are species, Plasmodium falciparum I want to look for.
Species.
Yeah,
So biological species, right?
yeah,
If
yeah,
you go and click here,
yeah.
and then you can select now.
Hmm.
It is organism,
Okay.
prokaryotes, and unclassified organisms. So if it is bacterium, eukaryote, virus. So is it bacterium?
It's a eukaryote.
Eukaryote. Okay.
Yeah.
Uh,
You can directly enter in the search term Plasmodium. It will tell you.
okay.
Yeah, yeah. Plasmodium. Yeah.
DM is not coming actually. Yeah,
Okay.
it is coming. Plasmodium. Let's
Uh,
say plasmodium.
Yeah.
Let's select Plasmodium. And transfer. And array something else happened. So I will type Plasmodium itself, Plasmodium. And what we'll do, we'll put a star there. So
Okay.
whatever is captured after the Plasmodium falciparum you are seeing, it will get captured. Now, if you want to have understand related documents, you can go and search the documents related to Plasmodium and now it will have Plasmodium falciparum as well embedded into it.
Okay.
Then you can,
I think all platform data will come here.
everything. And then you can, based on the Reaxys tree, you can look at the properties information, or based on the index term itself, you can actually see what you are looking at it in terms of the activity, toxicity, whatever related to this compound. You can switch to the substance information directly. You can based on, say, you are not looking at a patent information, so you can zero down based on what article in journal article, review article, anything you are, if you want to sort it down, you can do those information also, year based information. If you are aware of any author working in that particular area, you can do that.
Yes,
or you can now these 4,000 documents have 59 almost 60,000 substances and 400 targets, so you can modulate directly, go to those targets information also, right? And then find out the related information there. And here you go, there is a UniProt information directly, where you can get the protein or the particular target information directly in the external database. And related compounds.
Sorry to interfere, sir. I think we have some queries. If Nishant, it will be appreciated if you can contact Mandar sir.
Yes.
You can discuss your issue in detail separately, because some more are waiting.
And so this this webinar will be recording will be available for future reference.
Yes, I think they have recorded, so I will also like to have the recording.
Yeah. So we have one question from Mr.
Yes.
Nishit Araya. Nishit, please go ahead with your question. Nishit, please go ahead with your question, Nishit. Nishit, he is unmuted. Nishit, please ask the question. Okay. Nishit, are you there? Okay, sir. Okay, we have Rahul K. Rahul K, please unmute yourself and ask the question. Hello, Rahul K. Nishit, are you there? Okay. I think no more queries, sir. So,
okay.
Is it anything to continue? Please, like, you can conclude your presentation.
Nothing. I will just again go back to my screen sharing of my contact info. If anybody wants to contact me directly or have questions, you can go and ask me there.
Yeah. So,
or
Convener,
write an email.
has one question, please.
Who?
Hello.
Yes. Tell me.
Mandar, I have one question.
Yes.
How different is from SciFinder?
Yes. So it's a very nice question. So
equally competitor.
Yes. So what I think, have you used SciFinder completely, or are you using it?
I used it in 2007-8 in Canada.
Oh, okay. So if you see, I have shown you the drawing features, right? So one biggest feature is the ChemDraw inbuilt in this Reaxys, first of all, where SciFinder doesn't have it. We have text to structure drawing, which SciFinder doesn't have it. Then the natural language programming, the text-based search, when you type 2-3 letters, it will show you options. That is one of the difference. Then the query builder, where you can do complex searches. SciFinder doesn't have it. Then the the target information or the biology related information or the pharmacokinetic information, SciFinder doesn't have it the way we show it.
There are so many advantages.
Yes, we don't have structure activity relationship. We can do it. Also, for every compound, you can get the data directly in front of you in one place. You can directly search HPLC data, NMR data, IR data, UV visible data, as I showed you directly, right? Where it's not possible to get that information in SciFinder. So there are multiple advantages of this product over SciFinder. Also, 99% of it is cheaper than SciFinder also.
And user friendly also.
Yes, certainly user friendly.
Now I'm using this Reaxys. not
Oh,
in our
okay.
college, but I'm going to Indian School of Science, Bangalore.
Oh, okay, okay, okay. Yeah, yeah, they have it.
I'm accessing there.
Okay,
Now
great.
we have one more question from
Yes.
Shankar Aligaon.
Okay.
Shankar,
Yes, sir.
please ask your
Hello.
Yes.
question.
Yes, sir.
Sir, can you hear me?
Yes, certainly.
Yeah, beautiful presentation, Dr. Mandar.
Thank you.
Okay. Actually, Karki sir has told same question for myself also. Okay. So, what is there any evaluation so we can get it? So, because Science Direct were from last 6 to 7 consecutive year we are using, we are
Okay.
paying almost 5 lakh to Science Direct every
Okay.
year from KLE College of KLE Institute.
Okay.
Okay. So if you give the some evaluation and other things, it is very easy to put forward in meetings and other things.
No, certainly we can. I can work it out. I will get my management approval. So 15 days, up to 15 days, I can certainly get it. But I will try my best if I can provide you a little bit more time also for evaluation. But up to 15 days, I will try to get my management approval for sure. And
Okay.
give me some time, and I will get back to Dr. Karki also. And
So
once.
what about coverage, Mandar? Because if you see SciFinder, it is especially in ACS. Okay. If you see now, whatever you are told, it is the most about in other than this one, JAX. Okay.
No,
American
no.
Chemical Society.
So we cover 16,000 journals. So if you want to,
No, whether that include American Chemical Society,
yes. So
whatever the
yeah,
SciFinder.
I will just take you there. So if you go to Query Builder there, I hope you are able to see my Reaxys page, and you type
Yeah.
journal title. I have not shown so many things because the time was so short to show. So you can
No,
type
it is
journal
very good.
journal
Okay.
title, and now you want to see JAX, right? Journal of American Chemical Society.
No, related. American Chemical Society, jo, no,
You tell me
so
any
you
journal.
can,
So you, if you want to know, you can.
Okay.
sir, JMC, Journal of Medicinal Chemistry.
Right, JMC. Now if you see Journal of Medical Chemistry, you go to documents, you will come, you will have all the documents and all the publications coming from JMC, right?
Okay.
So there are 39,506 document. You can go publication year document, then you can sort it down whatever you want. So if you are worried about, you can actually do that. You can go to the query builder, type journal title, and
Yeah.
then you can type the journal name, you will get it. We have 16,000 journals coverage in Reaxys.
Okay. Okay. So what about Royal Society? That also is covered,
Yes.
sir?
Yes, sir.
Okay.
Everything is covered. So you want to have chemical communication, Chemcom?
Yeah, chemical communication or
Tell me name.
okay.
RS advances.
Yeah,
Yeah.
it is there. It is actually open access journal. So you will
Yeah.
obviously have. See, RS's
No,
advances.
earlier it was there.
We
Now
have to
some
press for clicks.
companies.
RSA advances documents. You will
Yeah.
have that. 58,730 documents of those. Again, see, the beauty of this is that you have substances captured within those documents. You have reactions captured here, targets also captured here, right? Similar way, you can go to the patent. I have not even captured the patent. Just quickly, I can, if you have time, I can show you that information also. Say patent inventor. You can say inventor name. You can type say or patent information. If you want, you can say patent. Location in patent, graphic, patent assignee, specific, all this related to country code. Let's do the country code. And I was talking about the Chinese patents. So let's type Chinese and see if we are able to get the information translated in English. Right? So see, the abstract will be in English. The claims will be in English. If you see English translated. Let me find out any chemistry related information patent, because I've just typed CN, right? So quickly, if you are able to find any chemistry information. No, yes, no, yes. Okay, see here, here you go, substances, right? So whatever the substances captured in the patent, you do not have to even go and see the patent. It will be there here in itself in front of you, right? Reactions, there are 13 reactions for this in the captured patent. You will have that information directly there. Reactions, if you go there, reactions, see, exactly captured here in the patent. So you have procedures, experimental procedures, you have claims, you have abstract, you have front page info, everything in translated form, right? You see, go here, conditions. Let's see if you click here, experimental procedure. See here, right? you have English. So this is a Chinese patent, but you have the related information required for a chemist to perform the reaction in English. So you do not have to go and beat the bush to find the translator, get the procedures there directly, right? Similar way, it can be done for all the patents as well. Now, if I go back to and if you want to search based on the inventor, so you can do those based on inventor, author searches, everything you can do it. Patent inventor. If you want to name any, if you have a patent, let me know, or just let me type my name just to showcase and then show the documents. Again, the name can be differently. So if I type another name, only write Bodas Mandar, you will see more patents coming up there. And here you go, the patent information, compounds captured, reactions captured, everything there. And the beauty is that if you want to analyze the patent, all the compounds together, select that, create a heat map, create a SAR of that compound directly. So what targets, what are the best compounds, everything in one place you can see there. So all the 1,000 compounds in the patents can be actually analyzed quickly. This is a beauty of Reaxys, right?
I thought Shankar sir got his answer.
Huh?
Hello, Shankar sir, is it clear?
Hello.
Yes.
Yeah, thank you once again, Mandar and Karki sir, Sujit, KLE College of Pharmacy, Bangalore, for organizing a very beautiful, very informative webinar.
Thank you.
Yeah, thank you, sir. And we are in future also we are looking for such a beautiful webinar, informative webinars. Okay.
Certainly.
Yes.
Thank you, sir. Thank you.
So this was the last question we took from the participants, and Mandar sir has just shared his screen, his contacts, mail ID. If anyone further wants to clarify anything, please contact him personally and get benefited from the tools what he has. For your research. So with this one, I like to thank the speaker, Professor Mandar, Mr. Mandar Bodas, and all participants on behalf of our whole KLE team, my colleagues, and especially on behalf of our BSRC team of campus in Bangalore. So thank you very much, sir. You managed to give such informative stuff in this tense situation. Everyone is worried about COVID. Hope the information we got here might be used for the future research of the researchers and participants. With that, once again, thank you very much to one and all present here. Thank you very much, sir. I am now concluding this session.
With that, once again, thank you very much to the session.
Thanks a lot.
Thank you, sir. Welcome, sir. Thank you, everyone.
So thank you again for providing the opportunity for me to present as well.
Thank you. Thank you. Thank you, Mother.
Yeah.
We'll keep in touch. Okay.
Yeah, certainly. We'll get back to you. Yeah.
Yeah, sure. Thank you.
Have a nice day, sir. All of you.
Thank you. Take care, sir. Bye.
Bye.
Take care and be safe.
Yeah, bye.
Okay, bye.